Hello everyone, and welcome to our first Royal Free Charity Presents free speech event. My name is John Spires. I'm the Chief Executive of the Royal Free Charity. The Royal Free Charity exists to help our amazing NHS heroes go further and do more so that our patients get the best possible care. We do that by funding equipment, by funding improvements to our hospitals, and crucially by funding life-saving and life-changing research. Many of you will have seen a snapshot of what goes on across our three hospitals, thanks to the incredible BBC documentary Hospital. While we can't quite match BBC production values, these free speech events aim to give you a regular glimpse of what goes on here at the Royal Free London NHS Foundation Trust. And each month we'll be bringing you a different speaker to give you different insights into the clinical and research activities that go on. Tonight's speaker promises to be very special indeed. I'm very pleased that we're going to be joined this evening by Professor Emma Morris. Emma is the Professor of Clinical Cell and Gene Therapy at the UCL Institute of Immunity and Transplantation, and she's an honorary consultant haematologist at UCLH and at the Royal Free. She developed and leads the National Stem Cell Transplant Centre for adults with primary immune deficiencies, and she works on gene therapy and gene editing techniques to develop new patient treatments, which hopefully we're going to hear about this evening. After Emma's talk, we'll be joined by Dr. Chris Strether, who's the Chief Medical Officer for the Royal Free NHS Foundation Trust. And Chris will be passing questions on from you, which you can put in the chat or in the comment stream to Emma. And we'll be finishing in around about 45 minutes. That's all from me until the end. So Professor Morris, over to you.
Thank you very, very much, John. I just want to say welcome to everyone. Thank you for giving up your Monday evening. Hello to my parents if you're there. This has to make do instead of a Christmas get-together, but at least you're spared my terrible cooking. But seriously, it's a great honour to have been invited to give the inaugural free speech supported by the extraordinary Royal Free Charity. The title of my talk this evening is Correcting Nature's Spelling Mistakes. I will describe to you how doctors and scientists at UCL and the Royal Free are developing exciting new treatments using stem cells and gene therapy for patients with serious inherited diseases. I'm going to start with a basic biology lesson. It's possible, given the amount of science, medicine, virology, and immunology that's been discussed in the newspapers, on television, in social media this year, that actually you won't need it. But I'm going to do it anyway. Every single one of us is unique. We are different physically and we're different psychologically. Most of us look different to each other too. However, within families, I'm just going to find my first picture. Within families, this is where I thought we might have a problem with my IT. I can do fancy gene therapy, but, oh dear. Let me just, there we go. Can you see my slide? That's really embarrassing, isn't it? Okay, so I can edit your genome, but I can't share a PowerPoint slide. It's a bit like giving a talk muted on Teams. Anyway, I'll carry on. Within families, we're much more similar. This is partly due to our shared experiences, which we call nurture, but it's also due to nature, our genes. So there's no guessing whose daughter this is a picture of. Hi, Ellie, if you're watching. Humans are complex machines. Each adult is made up of 37 trillion cells. That's 37,000 billion cells. Each cell in the body does a different job, with every organ of the body having its own very specialist cells. For example, cells of the heart, when you grow them in the dish, actually contract. They're programmed to do this. Many nerve and brain cells have long tendrils which reach out and touch each other. in order that chemical messages can be passed between the neurons. Cells of the immune system, however, can change shape and move between tissues and secrete poisons to kill dangerous cells. These are just a few examples. But in turn, each individual cell is made up of billions of protein molecules. So if you think of an analogy, think of a cell in your body as being a house. The proteins are all the things in the house that are useful and do stuff. So they make things work. For example, internal partition walls, staircases, electricity, water, furniture, kitchen appliances. There is one room in the house that is really special, and in the cell, it's called the nucleus, and that's the purple bit in the middle of this picture. And this contains our genetic material, which is basically the equivalent of the detailed architect's drawings for the house. Or trying another analogy, the script for a play with all the producers and directors' instructions. So, one of the interesting things is that every single day, our bodies need to continuously generate new cells, lots of them. And this is absolutely crucial because approximately 1 million cells die every second. So when you think about that, that's about 1.2 kilograms worth of cells in your body dying every day. Now, obviously, without replacing those dying cells, we wouldn't survive. When a cell divides, can you see that? Oh, I'm not very good at switching between these, am I? Here we go. Can you see that now? I'll be good at this by the end. When a cell divides, it generates two identical daughter cells. This is a complex process, but it's beautifully elegant and made possible by the fact that our genetic material, which is packaged in these funny chromosomes, those are the blue and red sort of pairs, the DNA or the genetic material packaged in that takes the form of a double helix. Well, what's a double helix? James Watson and Francis Crick, with vital help from Rosalind Franklin and Maurice Wilkins, discovered this three-dimensional structure of DNA way back in 1953. And their key observations were that DNA was double-stranded, a bit like a zip, and that the strands were complementary. So their strands matched each other perfectly. They finished their scientific manuscript way back in 1953 with the sentence, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material." The copying bit is really, really important, and we'll come back to it. Watson, Crick, and Wilkins jointly won the Nobel Prize in Physiology or Medicine. I think, appallingly, Wilkins' female colleague, Rosalind Franklin, did not receive the prize despite her major contributions. And sadly, she died just 5 years later of cancer at the age of 37, without seeing the impact of her discovery. Fast forward nearly 70 years, and we know now that if you unravel all the DNA within the nucleus of a cell, it stretches out to measure 6 feet in length. 6 feet, just the DNA in the nucleus of one tiny cell, and you have 37 trillion cells in your body. That is a lot of genetic material in your body. This DNA is divided into segments that we call genes, and each individual gene contains the code for a protein. This code is written in 4 letters, A, T, G, and C, and we call them bases or nucleotides. When a cell divides, DNA is copied to DNA. This ensures that daughter cells have identical genetic material. They can therefore become a true replacement cell and perform the same job in the body. So imagine it's like an insurance claim, like for like. It's okay for our children to be a little bit different to us, but actually, if one of your liver cells dies, you want it to be replaced by a liver cell. You don't want it to be replaced by a heart cell or a skin cell. So it's really important that the daughter cells are the same as the parental cell. So when a cell needs to make new proteins, parts of the DNA code, so that's on the left of this picture, are converted into a temporary messenger called RNA. Now, this RNA acts as a kind of template for the construction of proteins. And remember, proteins are the things in your cells that do stuff, things in your body that do stuff. For example, receptors, enzymes, growth factors, hormones, clotting factors, antibodies. The genes don't do anything, the genes are the instructions. This cartoon is drawn by a friend of mine, a very talented scientist called Pedro Vellica, who used to work here at the Royal Free in the Institute of Immunity and Transplantation. And what it shows in blue, that strand with the lumps on it, is RNA. And it moves from the nucleus into the cell, the inside of the cell, and it goes through a brown blob, which is called a ribosome. And whilst it passes through that brown blob, the template, the RNA, gets converted into a protein, and that's those grey blobby things coming off the top. So that's how RNA is turned into protein. So this cartoon actually is explaining how the new BioNTech Pfizer COVID-19 RNA vaccine works. And we started vaccinating people here at the Royal Free this last week. So the pink strand in this cartoon is actually RNA, which codes for a protein fragment, which is part of the outer coat of the coronavirus that causes COVID-19. When this pink RNA is taken up by our cells, it's not pink, by the way, but it is in this cartoon. For example, after you've had the vaccination, your cells dutifully make the harmless coronavirus protein fragments and trick the body into thinking that it has been infected. And so an immune response is triggered, which basically means you've made an army of soldiers specifically able to recognize and kill the virus. And then they lie in wait and protect you by preventing the virus, if you did get infected by contact with someone, from surviving long enough in your body to become ill. You'll be pleased to know that I'm nearly done with the biology refresher. The Human Genome Project was an international collaboration of scientists working together with the sole aim of deciphering the entire human genetic code. It took 13 years, and cost 2.7 billion US dollars. But as a result, we now understand that every person has around 30,000 separate genes within their DNA. Most genes are only switched on in some specific cell types and for a specific time in the cell's life or the cell's development. For example, when an immune cell recognizes a virally infected cell, it may very rapidly need to make lots of toxic molecules which it can release to attack the cell harboring the virus. But actually, once the virus is gone, it needs to switch this system off so that the immune cell doesn't go around causing more damage in the body. So these genes get switched off again. So I'm going to summarize our biology lesson. Basically, as human beings, we are made up of DNA, which is the instructions for what can be made, RNA, which is like a recipe, what will be made, proteins, the product itself, here the example is a cupcake, and cells and tissues, which are a collection of products altogether. So instead of thinking ourselves as being DNA, RNA, proteins, cells, tissues, and organs, we're basically cookbooks, recipes, cupcakes, and fancy teas. So now you are all experts in genetics and biology. I'm going to explain how understanding genetics and basic cell biology has allowed some of us at the Royal Free develop new treatments for both rare inherited diseases and more common diseases like cancer. Most diseases are what we call acquired, like influenza, which is caused by the flu virus, or lung cancer, which is most frequently caused by smoking, or liver cirrhosis, which can be caused by alcohol. However, other diseases are referred to as genetic or inherited diseases, and they're caused by a mistake in the person's DNA. Genetic conditions are usually inherited, meaning they're passed down from parents to their children. But some occur in individuals when there is no family history of the disorder. In these cases, the genetic mutations or genetic mistakes occur during the development of the egg, the sperm, or the embryo. Genetic mutations can result in one or more genes being deleted or rubbed out from the code book. If this gene was one that gives the instructions to make a particularly important protein, such as a blood clotting factor, people born with this genetic mistake will not be able to make that clotting factor because the code has been rubbed out. They will then suffer potentially from spontaneous bleeding, which can be life-threatening, as in haemophilia. Other genetic mutations, which we can call spelling mistakes in the code rather than rubbings out, they scramble the letters so that the code no longer makes any sense. And in such cases, the cell just can't follow the instructions, and the protein is either not made or only part of the protein is made and it doesn't work properly. Some genetic diseases are caused by mutations that make excessive amounts of protein or an overactive variant of the protein, and we call these gain of function mutations. I won't go on forever, but sometimes it matters a lot whether you carry one or two genetic mistakes. Apart from genes that are found on the X chromosome, we all have two copies of every other gene, one from our mother and one from our father. In some diseases, you can be completely well, even if you have one mutation. but you're still able to pass it on to your children. We call this being a carrier. So now we're going to move to the exciting part. Exciting because I'm going to try and share my screen again. Has that worked? Ha! I've nailed it. It's brilliant. Okay, so the dramatic increase. Oh, I haven't. Sorry, I spoke too soon. The dramatic increase in knowledge regarding genetics has resulted in the development of laboratory tools for genetic engineering. We can cut DNA precisely, amplify sections of it, insert new letters, copy it, and even repair mistakes in it, all in the laboratory. It's a bit like having an advanced word processing software at our disposal. And as a result, we can now do two things. We can correct genetic mistakes in patients, thereby providing a permanent cure for the disease. And we can use genetic engineering to improve the function of other cells. For example, generating highly armed immune cells able to recognize and kill cancer cells, which are not otherwise responding to chemotherapy or radiotherapy. These treatments are referred to as gene therapy, gene editing, and cell therapy. Gene therapy is any treatment that introduces an extra copy of a healthy gene into a cell. I'm going to use a kidney analogy now because lots of the senior staff at the Royal Free happen to be nephrologists, which is kidney doctors, and Chris Strether is one of them. So this form of gene therapy, where you just add a healthy copy to the cell, is a bit like doing a kidney transplant. So surgeons tend to leave the non-functioning kidneys in the body and just, in inverted commas, plumb in a new one. The body then uses the new functioning kidney to get rid of toxic chemicals in our body, control the composition of salts and acids, and even make urine. But they leave behind the shriveled up kidneys that aren't working properly. So this form of gene therapy can be used to replace missing genes or genes that don't function properly. Patients with severe immune deficiencies can die in their very first year of life if their abnormal immune system is not very quickly replaced by a healthy immune system. And the healthy immune system mustn't have any genetic mistakes in it. This can be achieved by bone marrow transplantation or stem cell transplantation. In this, the patient is given powerful chemotherapy to destroy their own non-functioning immune cells. But it only works if a donor can be found with matching healthy immune system. Before bone marrow transplantation was established as a treatment for severe immune deficiencies caused by genetic mutations in immune genes, the only alternatives at the time were to remain in isolation. So there was no risk of exposure to infection of any kind. And you'll see here a picture of a boy called David Vetter, who was born with a form of severe combined immune deficiency in 1971. And we think we've had a tough time shielding some of us this year because of COVID-19. But this boy lived in a bubble for 12 years with no physical contact with his family, his brothers and sisters or his parents, because they couldn't find a matching bone marrow donor for him. Eventually one was found for him, but sadly he died of transplant complications a year later when he was only 13 years old. Here at the Royal Free in the Department of Immunology, we provide specialist clinical services for the largest group of adolescents and adult patients in Europe with various forms of severe immune deficiencies. Some of these patients were the very first in the entire world to be treated successfully with gene therapy when they were babies at Great Ormond Street Hospital. As they did not have matching bone marrow donors, gene therapy was used for the first ever time. Their own bone marrow stem cells were removed and genetically engineered in the lab. And by this, I mean a healthy copy of the faulty gene was introduced into their stem cells, and then the corrected stem cells were reinfused back into the patient, where they produced healthy blood cells and healthy immune cells. This worked. It was the first proof that gene therapy could actually work in patients. And it works so well that these boys have now, these trailblazers in the last 2 years, have transitioned, they've grown up, they've lived long enough to transition to adult services. And I now have the privilege of looking after them and monitoring them for the long-term efficacy of this stem cell gene therapy, where they come to clinic at the Royal Free. And I think it's fair to say both them and us are in uncharted waters. At the same time, here at the Royal Free, we have been the first hospital in the world to use the same forms of gene therapy, but not in babies, but in adults, to successfully treat other forms of immune deficiencies, creating treatment options for patients where there are no other possibilities of treatment. As I said before, these approaches rely on giving patients an extra healthy gene or simple gene addition. But for diseases where the mutated gene produces a protein which either damages the cell or is overactive and dangerous, giving an extra healthy copy will not correct the problem, as the bad or overactive protein will still be produced at the same time. So it's not like a shriveled up kidney that's not causing any problems. For these types of genetic disease, we need to be even more skillful and try to actually repair the genetic mistake. This is called gene editing, which scientists in my lab are developing for other immune deficiencies. This brings us right up to date. As we use gene editing technologies referred to as CRISPR, this was discovered by 2 women scientists who were acknowledged for their outstanding research. This is Jennifer Doudna and Emmanuelle Charpentier, who were awarded the Nobel Prize in Chemistry this year in 2020. I'm going to close by mentioning the pioneering work being done at the Royal Free in developing immunotherapies for cancer. Genetic engineering can also be used to improve the ability of a cancer patient's own immune cells to recognize and kill cancer cells. And these can be cancer cells left behind after treatment with chemotherapy. This can either be achieved by equipping the immune cell with additional weapons, if you think of the immune cell as a soldier against cancer, or by helping the immune cells to actually recognize the cancer cells more efficiently. Sometimes they can't tell. Immune cells find it hard to tell a cancer cell apart from a normal cell, or they just ignore it. These particular immune cells are called engineered T cells, and the most commonly used are CAR T cells. They were first given to children with leukemia in 2012, so only 8 years ago. And this was done in Philadelphia in the US. Emily Whitehead, shown here, was on her deathbed with only days to live, having not responded to any other treatments. This year, she marked 8 years completely well and leukemia-free, having had a single dose of genetically engineered immune cells that were able to recognize and kill her leukemia cells. This scan is called a CT PET scan, and the red blobs are metabolically active tissue. So for example, that can be your heart or your brain, but all the other red blobs are tumor cells that are growing really aggressively in the body. And what you can see here, again, with a different type of blood cancer, a single dose of engineered T cells can kill all of that tumor in the picture on the left, the red blob up around the neck. And 10 months later, the only red left behind is the heart and the kidneys and the brain, which in healthy people are supposed to be metabolically active. So the Royal Free, the connection to this is that the Royal Free has its own laboratories that manufacture CAR T cells and other gene therapies for patients all across the country who are on clinical trials with CAR T cells. So I think that 2020 has been an extraordinary year, maybe more now than ever, maybe in the entire history of the NHS. We have understood how remarkable our hospitals are and the people who work in them, and how we as individuals quite literally owe our lives to medical progress and to science. So I want to thank you for giving up your time. Thank you for being bamboozled with some science. And thank you for your support of the Royal Free Hospital and the Royal Free Charity.
Brilliant, thanks Emma, that's fantastic. I was at medical school quite a long time ago. I went in 1981, but some of this stuff is just stuff that was science fiction when I was at medical school. The idea that you give a single injection of a gene and someone 8 years later looks as healthy as the little girl you showed is just wondrous to me. I've got a couple of questions and I'll solicit a few more. So I'll give you the first two questions, Emma, and then we'll solicit a couple more. So the first one, I'll give you a group of two. So the first one's from Marie Gray. It's, can gene therapy be used to correct essential thrombocythemia?
Oh,
okay, why don't you do that one and then we'll do the second one. So do the first question.
Sorry. Yeah, for those people who don't know, essential thrombocythemia is a blood disorder where the bone marrow produces too many platelet cells, and the platelet cells are the cells in our blood that form blood clots. And patients with ET often have a genetic mutation in the stem cells in their bone marrow. In answer to your question, at the moment, there are no preclinical trials exploring gene therapy for patients with essential thrombocythemia. It's not inconceivable that they can't be developed in the future, but it's one of those diseases where the genetic factors also, there are also non-genetic factors that help with the progression of the disease, which can't always be corrected by gene therapy.
Thanks, Emma. So I'll ask the second question. Second question is from Helena Hailu. It's, Emma, could you please comment on gene therapy for type 1 diabetes at an early stage of diagnosis?
Oh, now that's a great question. So the way in which gene therapy currently is being developed for type 1 diabetes. So for people who don't know about type 1 diabetes, this is a bit of a crash course in all medicine for me. I hope, well, anyway, you know, as my husband would say, you just be confident and make it up. I don't think I'm making this up. Type 1 diabetes is an autoimmune disease where the immune system, your own immune system, attacks parts of your pancreas, and your pancreas produces insulin, which controls the level of sugar in your circulation. And if the levels of sugar aren't maintained at a very level concentration, you can end up with lots of tissue damage. You have problems with blood pressure, you can have problems with your eyesight, you can have kidney disease, you can have problems with your blood vessels, and it's a very serious disease. Now, gene therapy is being used, but it's being used in a slightly different way. So researchers are trying to develop immune cells that aren't killer immune cells, but that are suppressor immune cells, or rather regulatory T cells, which have the ability to actually switch off immune responses. So these are ways of genetically engineering immune cells, which if you inject them into a patient, in theory, would travel around the body, get to the pancreas where there's all the inflammation going on and tissue damage caused by overactive immune cells. And these genetically engineered suppressor immune cells will switch off that inflammation and immune overactivity. And there are a number of early phase clinical trials in the pipeline. They're not open yet, but there's a number of groups in London and in the US trying to develop these approaches for type 1 diabetes. So that's a way of using gene therapy by genetically engineering immune cells, but instead of making killer immune cells, making powerful immune suppressing cells.
Thanks, Emma. I was looking at that question thinking I was very glad I wasn't being asked it. The next question is from Farhan Naim, who's our director for research and development. So he thanks you for the fascinating lecture. He said, what have been the early results of CRISPR, CRISPR in cancer, and how far are we away from human trials? He's got the advantage that he understands the question and I don't. So Emma.
So I think it's a good question, Farhan. CRISPR is the gene editing technology that won the Nobel Prize this year. So these are like genetic scissors that allow you to repair genetic mistakes inside a strand of DNA. In cancer, again, CRISPR is being used to actually improve the function of immune cells that are being given to patients to attack the cancer cells. So at the moment, there are a number of trials planned which will not only use immune cells that have new genes for new receptors added to them, but they will have various proteins knocked out by the CRISPR scissors or genes cut out, which would make the immune cells less effective over time. So these are in the pipeline and early phase trials are proceeding in the US. So it's a bit early, Farhan. I'll have to come back in 5 years' time.
Thanks, Emma. I've got a question. While we're waiting to see if anyone else has a question, I've got one. I remember hearing you present about some of this before, and hearing Pratima talking about some of the trials in haemophilia, and with some of these inherited diseases, as people get older, they get other, I suppose, kind of end organ complications of the disease and become frailer, to be honest. And there's sometimes a balance to be struck, but about how early you give a treatment. You know, if you give the treatment very, very early, before people have got other complications, then you give them a longer, healthier lifespan. But the unknown about whether, you know, the long-term effects, then they've got a longer lifespan ahead of them to tolerate those. So, and I think that's a really difficult kind of, I suppose, philosophical question. But how do you approach that question about balance, about when's the time to intervene?
So it's, in practice, it's really difficult, because when you're using treatments that are really quite experimental, so they haven't been used on many people and they haven't been used for very long, so we don't understand whether there are potentially long-term side effects or complications, and also we don't know how long the beneficial effect of them is going to last. There is a real, when you talk to patients about these sort of treatments, you have to develop, you can't do this without having developed a really honest relationship with the patient and their family. And in most cases, we would always wait until the patient has developed the sort of complications that we know for certain are likely to cause more and more serious problems over time. So if we're able to identify particular characteristics of the disease or particular complications that patients have had, by which we know that actually in 5 years' time they're going to be really, really unwell, then we know that it's not an unreasonable thing to intervene with a treatment that they may benefit from, even if there are a number of unanswered questions. Clearly, if there are other existing standard treatments that are working, you would continue with those. But for many patients, they've tried all of those and they're not working. Now, the reality of it, in practice, that's brilliant if you can always get someone in that sweet spot when they're still well enough to have the treatment, but you know they're going to become seriously ill and they're most likely to benefit from it. But actually, sometimes what happens with diseases is that these rarer inherited diseases, that people are reasonably well and then all of a sudden they get very, very unwell, and by which time it is too late to intervene. And this is why there's quite a lot of research in the field to try and work out what we call biomarkers, but they're basically predictors of whether people are going to become more unwell. So for example, if you come to hospital and you're asked to join a research project where we maybe do x-rays a couple of times a year or take blood from you, and you think, well, what's that all about? Often it's about that. It's about us trying to understand the natural history, particularly for patients who have really rare diseases where there are not very many people around the world with them.
Thanks, Emma. We've got about 4 minutes left, and I've got two more questions. So the first one is from Nicola Whitehill. So she said it's very encouraging for a patient with SSC, and thank you for all you've done. She says, are there any potential gene therapy developments for scleroderma?
Okay, so as far as I know at the moment there aren't, but we have recently started using stem cell transplants for patients with scleroderma, and we do that in collaboration with Professor Chris Denton at the Royal Free. And there are a few patients with scleroderma who we are now recommending that they proceed to stem cell transplant or bone marrow transplant. And we use that as a way to try and reset their immune system. And that can be effective in a number of patients, but there are fairly particular inclusion criteria for that. At the moment, the problem with gene therapy for scleroderma is some of the genetic abnormalities are very widespread in the body and they're quite difficult to target. So if the genes that are abnormal, or if there are immune responses to many genes expressed in lots of different types of cells, you can actually unwittingly damage cells rather than just targeting the treatment very specifically.
Thanks, Emma. I'm very impressed by the breadth of your knowledge, because the questions, there's a lot of variety in the questions. So I think probably the last question before we sum up is it's a follow-up question from Marie Gray, who asked the first question. So she was talking about the combined immunodeficiency rarely comes alone, and she said, would it be possible to anticipate what other conditions could be lurking and possibly corrected by the process you've described in your great presentation? So I suppose kind of what's next in this vulnerable group of patients?
Sorry, I missed the very first beginning of that. Was it about patients with
multiple conditions?
Oh, okay.
So
I think,
yeah.
What a question to finish on. Basically, most diseases, so what we're talking about here actually, are very rare inherited diseases where you have a very clearly defined one gene that's abnormal. So it's easy, in inverted commas, to fix that gene. And we're also talking about diseases like cancer, where actually you can genetically engineer an immune attack against the cancer. But for all sorts of other diseases like chronic lung disease, liver disease, kidney disease, most of these are caused by many, many different factors. They're what we call either polygenic, so lots of different genes are involved, so you might have a tendency to get these diseases, but there are also environmental effects, there are dietary effects, they could be infectious elements to it. And actually, in the future, trying to work out whether gene therapy can tackle any or all of those will be the big challenge.
Good. Thanks, Emma. I think we have to draw it to close because it is now 7:15. But I'd like to thank Emma for one, a brilliant talk, but two, an incredible adeptness at answering a variety of questions. At least one of the 6 questions I didn't understand. So thank you very, very much to. I didn't understand because it was too sophisticated, not because it was incomprehensible. And so thank you very, very much to Emma. I don't know if John wants to say any closing words, but thank you very much to Emma. John.
Thank you, Chris, and thank you from me as well to Professor Morris. That was an absolutely fascinating talk, and thank you to everyone who's joined us today. My only message is don't forget to tune in this evening on BBC 2 at 9 o'clock for the final episode of Hospital. If that whets your appetite for more, then we'll soon be announcing details of the next RFC Presents Free Speech event, which will be sharing the views and experiences of some of the faces of the documentary. So thank you very much again, everybody.